Our last dataset, the IRIS dataset, is used to demonstrate various classification machine learning algorithms. There are three species of iris flowers: the setosa, the versicolor, and the iris virginica. The dataset contains information about 150 iris flowers. For each flower, we have the sepal length and width, the petal length and width, and the species. And the species column here is called the class. So the idea is that from this dataset, a machine learning algorithm would learn to classify new unseen flowers based on these features, based on the sepal length and width and on the petal length and width. In machine learning, it's very useful to visualize our data when possible to see if that data is separable or if we need to do some extra work to make it separable. So let's see how to do that with our iris dataset. Let's first modify our main function to call explore iris. So here we read the iris file into the df flowers data frame, and we use class as our index column. We create three scatter plots. For the first one, we only select the iris setosas. We use log and specify iris setosa, and we plot the sepal length and the sepal width. So our x-axis is going to be the sepal length, and the y-axis is going to be the sepal width. For our second plot, we also plot the sepal width versus the sepal length, but we select the iris versicolors. And for our third scatter plot, we select the iris virginica. I just want to point out here that the rows are selected by specifying log. The label is just added to the plot at the end. Let's run our program. And here's our scatter plot. So the blue dots represent the iris setosas, the orange dots represent the iris versicolor, and the green dots represent the iris virginicas. We can see here that the iris setosas are separable from the other two species, but the iris versicolor and the iris virginica are not separable here. We can also plot our petal width against our petal length. We can do a quick change here. We change all sepal to petal. And let's run our program again. And here's our new scatter plot. So here, iris setosa is clearly separable from the rest. Iris versicolor and virginica are a little more separable than before. So here we were limited to 2 dimensions. We were able to plot 2 features, the petal width versus the petal length, or the sepal width versus the sepal length. But it's sometimes useful to plot 3 different features, and we can do that with 3D. So the first thing to do here is to uncomment this import. We need mplot3d, and then here's our code to plot a 3D figure here. We have to add these 2 statements here to create the figure and the axis. We specify 3 different series as arguments to the scatter function here instead of just 2. We have one series representing the x-axis, one series representing the y-axis, and one series representing the z-axis. And we label the 3 axes here. Let's run our program. And here's our beautiful 3D plot.